Welcome student. So in last lecture we have started the point packaging of DNA, and in that lecture we have seen that how there is a packaging of such a long DNA in prokaryotic cell. Now today we will see how such a long DNA is packed in a eukaryotic cell. Now in last lecture I have explained you the difference between prokaryotic cell and eukaryotic cell. Eukaryotic cell is much more bigger than the prokaryotic cell, and its level is also high than the prokaryotic cell, and it is of advanced type because from the prokaryotic cell the eukaryotic cell has been evolved. Now if we see in eukaryotic cell, if we take the calculations, then according to the calculation the DNA measures about 2.2 meter long. DNA measures about 2.2 meter long. Now how this possible? Because if you see our height is about near about 6 feet or 5.5 feet. Now if you see 2 meter means near about 6 feet. So the DNA fiber or DNA is long as our height. How such long DNA can be fitted in such a small size cell which is microscopic or which cannot be observed through our naked eyes? And this is done by the packaging. This is done by the packaging, and we are going to see this. Now if we take the nucleus or if you take the DNA, that DNA which is present in a nucleus is associated with histone and non-histone protein. DNA is associated with histone and non-histone proteins. These histone proteins, if we see, they are rich in lysine and arginine. These are amino acids. If you see, in last year we have seen the biomolecule chapter, and in which we have learned about the organic substances that are protein, lipids and carbohydrate. And if we have learned about the proteins in detail, and if you see the proteins, they are the polypeptide chain. They are made up of the polypeptide chains of amino acids. And in this amino acid, near about 20 amino acids are essential amino acids. And from this, we have seen lysine is also there, leucine is also there, arginine is there, aspartic acid is there, many much histamine is there, many 20 essential amino acids are present. So arginine and lysine, these amino acids are rich in this histone, lysine and arginine. Residues, their residues is present, and so due to their presence, these amino acids are positively charged or they are in basic in nature, and therefore these histone molecules are positively charged. They are positively charged. They are positively charged. And these histone molecules organize themselves in this DNA structure or in the nucleus. They organize themselves. Histone molecules organize themselves in together molecules of this histone comes together and form a core and forms a octamer. Octamer is 8. If you see, one is called as mono, 2 molecules di, 3 molecules tri, 4 molecules tetra, 5 molecules penta, 6 molecules hexa, 7 molecules hepto, 8 molecules octa, 9 molecules nano, 10 molecules deci. So such names are given. So when 8 molecules comes together and form a bunch, it forms a one structure that is called as what? Octamer. So such 8 histone molecules which are rich in residues of arginine and lysine, they comes together and form a structure that is called as octamer. And in this this octamer is wrapped by DNA, which is negatively charged. So due to this charge, positive charge and negative charge, the DNA molecule is wrapped around that octamer. And this structure together is called as what? Nucleosome. Nucleosome. We will see by figure. Suppose these are the histone molecules. They come together. They are present in the nucleus. Like this, they come together. Then DNA is wrapped. This is again a histone molecule which is called as H1, and this is linker DNA. So 1, 2, 3, 4. In between this one molecule is found behind. This is 5, 6, 7, and 8. So 8 such histone molecules comes together and form a DNA core. This is the core here, the nucleosome core here, and the DNA, due to the charge, positive charge and negative charge, there is the effect, and this DNA is wrapped. This is the wrapped DNA. Around the histone molecule, that due to the positive and negative charge. And it also, by wrapping, after wrapping, it leaves from this nucleosome. And when it leaves, it is connected to again to the next nucleosome. So this DNA which leaves from here, that is called as linker DNA. That is called as what? Linker DNA. Means if we see the nucleosome consists of 8 molecules of histone that are H2A, then H2B, H3 and H4, and all these are present in pairs, means 2 H2A, 2 H2B, 2 H3 and 2 H4. In all there is total 8. So 8 octavers come together form a main core of this structure. Then DNA is wrapped around it. Here it is present H1 histone molecule which attaches which packs the DNA which enters the nucleosome and leaves the nucleosome. It holds it. H1 histone molecule and linker DNA. So this is the structure of nucleosome, and this structure is repeated. Means if you see, suppose this is I will draw it as a one ball. I will draw this nucleosome as suppose this is a nucleosome. Then this DNA used from a linker DNA. Again there is a nucleosome here attached. This linker DNA used here. Again there is a nucleosome. Then again there is a nucleosome. Again there is a nucleosome. Because if we see the DNA present in a eukaryotic cell consists of at least 200 base pairs. At least 200 base pairs. And this histone molecule is responsible for wrapping such a long DNA around it. Means the structure becomes less and less, and quantity is the same. So this structure looks like beads on string. String means what? A thread-like structure. Beads means what? A ball-like structure. Means on thread, when there is an arrangement of balls like this is called as what? Beads on string. And same is the structure here. So this is called as this arrangement of nucleosome in a DNA is like what? Beads on string. So this is the structure of nucleosome. And if you see, these nucleosome structures are repeated units of chromatin which looks like bead on string. When such six nucleosomes comes together, and forms a structure that is known as what? Solenoid. 6 such nucleosomes comes together and form one structure that is known as what? Solenoid. Then there is again supercoiling of this. When such nucleosomes come together, they coil. Again there is a coiling and recoiling of this, and there is a form like loop-like structure that is called as what? Chromatin fiber. Solenoid. Coils and recoils to form loop-like structure. That is called as what? Chromatin fiber. and in chromatin fiber again get coiled and condensed to form. When it get condensed at metaphase stage, and at metaphase stage these chromatin fibers form what? Chromosome. At metaphase stage, this chromatin fiber coils and recoils and get condensed to form chromosome. And we know what is chromosome. So this is a very important phenomenon, because if we see the formation of chromosome is very important, because if we see the division stages, that is mitosis and meiosis. In mitosis and meiosis there is a division of chromosomes, and from this division the maternal material, then paternal material, there is a exchange also. And if you see in mitosis, there is a equational division, because from one cell two cells are formed, and in the two cells the chromosome number is same. Chromosome number is same, maintained same. But when there is a meiosis, chromosome number is reduced to R, and when there is a reproduction, the chromosome number is regained. Chromosome number is regained means for reproduction process, sexual reproduction process, the reduction division is very important. Means meiosis is very important, and for meiosis and mitosis there is a need of chromosome, because if you see the division, mitosis, in which there is two main stages, that is karyokinesis and cytokinesis. In karyokinesis we have seen four stages, four phases, that is prophase, metaphase, anaphase and telophase. At this metaphase stage, these chromatin fibers coils and get condensed to form chromosome. So such is the nucleosome. In this nucleosome, main points which you have to fit in your mind or in your brain is that the nucleosome consists of histone molecules which are positively charged, and there are about 8 histone molecules coming together and form a compact structure, and they are positively charged due to the effect of the residue of lysine and arginine amino acids. Then around this octamer, the DNA is wrapped. DNA is wrapped, and why it is wrapped? Due to the negatively charged. It is wrapped around that nucleosome, and due to this wrapping, the DNA or the length of the DNA is what? It is not reduced, but the volume is reduced. Or it takes less volume. So such is the structure of nucleosome. Now we will see one point we have to see here. In higher levels in eukaryotic cell, non-histone protein has also role. Non-histone proteins has a role in it, and these non-histone proteins are called as NHC. This NHC, additional set of proteins, non-histone chromosomal protein. This NHC is non-histone chromosomal protein. For packaging of chromatin at higher level, these are at higher level. For packaging there is need of this NSC for packaging. So these non-histochromosomal proteins are also used in the packaging of DNA at higher level. Now we will see the next point, that is heterochromatin and euchromatin. What is heterochromatin and euchromatin? Heterochromatin first of all we will see heterochromatin. What is heterochromatin? So the condensed region of chromosome, during interphase and early prophase, that condensed region of chromosome is called as what? Heterochromatin. That condensed region is called as what? Heterochromatin. The condensed region of chromosome during interphase and prophase. That is called as what? Euchromatin. And that is called as what? Heterochromatin. Now this region stains dark. How to recognize it? When there is a stain added to that, the region which stains dark is called as what? Heterochromatin. And this region is DNA inactive region, genetically inactive region, Then it is also transcriptionally inactive. Now you will have a question, what is transcription? What is transcriptionally inactive? Because if you see what is transcription, the formation of RNA from DNA, that is called as what? Transcription. It is a long process. We have to see this part later on in this chapter. When there is a formation of RNA from DNA, that process is called as what? Transcription. Transcription means to copy. What is copy? RNA copies all the genetic structure or genetic information regarding protein synthesis from DNA. So that process is called as what? Transcription. So transcriptionally this part, this condensed part or this heterochromatin region is transcriptionally inactive. It is transcriptionally inactive, and heterochromatin is 2 to 3 times more rich in DNA. Means DNA content is large or is more than euchromatin. Because sometimes the question is asked, distinguish between euchromatin and heterochromatin. Then at that time we have to give one point that DNA content is more in heterochromatin and DNA content is less in euchromatin. So this is heterochromatin. Then what is euchromatin? Euchromatin, non-condensed region of chromosome. It is non-condensed region of chromosome. That is called as what? Hetero. It stains like, second point, if we compare it, it stains like. Then next point, genetically active. If we distinguish between these two. Genetically inactive. This is active. DNA content is less. Then transcriptionally it is also active. Transcriptionally active. Then DNA content is less than heterochromatic. DNA content is less than heterochromatic. DNA content is less than heterochromatic. So, heterochromatic and euchromatic, in which main point or which we see the euchromatin is main one, which is a non-content part of a region of chromosome, in which it stands like it is a genetically active region, transcriptionally active region, and DNA content is less than the heterochromatin. So this is the heterochromatin and euchromatin. Now we will see the next point. Now the next point is replication. Before studying the replication, you should know what is DNA. And in last year chapters we have seen the DNA. What is DNA? Now, if we see in cell nucleus is present, and in that nucleus it is made up of nucleic acid, and this nucleic acid consists of what? DNA and RNA. Now, what is this DNA? If we see, the long form of DNA is what? Deoxyribose nucleic acid. Deoxyribose nucleic acid. And if we see the constituents of DNA, they are three constituents, or the DNA are made up of what? Nucleotides. DNA is made up of what? Nucleotides. Many nucleotides comes together and form what? DNA. And one nucleotide is made up of three molecules. Which are they? One pentose sugar. Which pentose sugar? Deoxyribose sugar. Which pentose sugar? Deoxyribose sugar. Second molecule is phosphate molecule. Phosphate molecule. And third constituent is what? Nitrogen bases. Nitrogen bases. Now, these nitrogen bases are made up of what? Two constituents. Main constituents are there: purines and pyrimidines. Now this purines has again two types of nitrogen bases, that is adenine and guanine. Pyrimidines are again made up of two nitrogen bases that are called as what? Cytosine and thymine. These are all what? Constituent of nucleotide. One nucleotide consists of three molecules, that are sugar, deoxyribose sugar, phosphate molecule, nitrogen bases. Phosphate molecule and nitrogen bases. So, and if you see these nucleotides, 10 such nucleotides come together and form DNA. More than 10 nucleotides are present. And according to the number, these nucleotides names are given according to the presence of the nitrogen bases. If adenine is present in that particular nucleotide, then that nucleotide is called as what? Adenine nucleotide. If guanine is present, then that nucleotide is called as what? Guanine nucleotide. If cytosine is present, then cytosine nucleotide. If thymine is present, then thymine nucleotide. And if you see the structure of DNA, these nucleotides or this DNA consists of two strands. This DNA consists of two strands. And these strands are made up of what? Nucleotides. And these two strands, their direction is different. One runs upward and second runs downwards. Means they are anti-parallel and they are bounded by nitrogen bases. Here there is a presence of nitrogen bases. Two nitrogen bases here forms the bonds. Forms the bond. That is hydrogen bond. And between nucleotides or between the sugar and phosphate, there is also a bond present. That is called as what? Phosphodiester bond. Phosphodiester bond that is present. And between nitrogen base and sugar molecule, there is a bond present which is called as glycosidic bond. Which is called as what? Glycosidic bond. Because for attachment of this, there is a bond and that bonds are known accordingly. Hydrogen bond, glycosidic bond and phosphodiester bond. Means the backbone of this DNA is made up of what? Nitrogen bases. The side chains, side structure, these two strands, they are made up of what? Nitrogen base. They are made up of what? Phosphate and sugar. They are made up of what? Phosphate and sugar. So the things we have learned, which is a double helical structure. It is a double helical structure. We have seen this in the last year's lecture. So this is we have to recall such point because we are going to see replication. And before replication, these points are made because if you see in replication, there is a mirror copy formation or there is a duplication of DNA. Because if you see in mitosis, what happens in mitosis? From one cell, two cells are formed. The DNA content which is present here or the number of chromosomes which are present here, the number of this chromosome and this chromosome, parent cell chromosome is the same. How this happens? How the number is maintained? This number is maintained by the process which is known as replication. So, we will see the next point of replication in next lecture.